Have you ever, really paused to think about that hidden instruction manual inside you? The one dictating, well, pretty much everything. Your appearance, how your body works, maybe even what conditions you might be prone to. Yeah, I'm talking about your genome, your complete biological blueprint. It's this massive DNA library. And here's what's wild. Even the tiniest changes, super subtle variations in that manual, can have these huge ripple effects on your health. So today on the Deep Dive, we're jumping into something really exciting. It's where AI meets biology, our mission. To explore this brand new AI tool from Google DeepMind, it's called AlphaGenome, and it's designed to shed some serious light on that complex genetic instruction book, especially what happens when the instructions change, even slightly. We've got some cutting-edge research, some big announcements to unpack here. We'll dig into what AlphaGenome is, how it actually works, and why it's such a huge deal for understanding diseases and maybe, just maybe, finding new treatments. Okay, let's really explore this. So we've kind of set up the genome as this ultimate instruction manual in our cells, right?
It's honestly mind-bending that this code guides almost everything about us. But even knowing how vital it is, figuring out how the instructions are actually read, molecule by molecule, and maybe even trickier, what happens when you get these small DNA changes, these variations? That's still apparently one of biology's greatest mysteries. It's like having this ancient, profound text, but you can only decipher parts of it, right? The really subtle, critical bits are still fuzzy.
Exactly. And that mystery, that fuzziness, is precisely where AlphaGenome comes in. It's really poised to unravel some of that. It's a new AI tool, a powerful one. And what it does is predict with really impressive accuracy how just a single change, a single variant or mutation in our DNA sequence might impact all sorts of biological processes, the ones that control our genes.
It's like a super translator for those subtle bits we couldn't read before.
That's a great way to put it. Yeah, super translator. And this wasn't possible before, not really. It took some big technical steps forward, allowing the AI model to process these really long DNA sequences, and then importantly, outputting very detailed, high-resolution predictions about what that sequence is doing. It's not just a small improvement. It feels like a genuine leap in understanding how our genome actually works.
Okay, that's fascinating. So the genome's the manual. How does AlphaGenome actually read it? How did this AI peek inside and figure out what all those DNA letters mean? And crucially, like you said, what happens when they change?
Right. It's pretty clever, actually. So AlphaGenome takes a really long DNA sequence as its input. We're talking up to 1 million letters or base pairs. That's huge.
1 million? Wow.
Yeah. And from that massive chunk of DNA, it predicts thousands of different molecular properties, things that tell us about its regulatory activity. Now, to figure out the effect of a mutation, it does a comparison. It predicts the properties for the sequence with the mutation and then compares that to the prediction for the original unmutated sequence. That comparison gives it a score for the impact of that specific change.
Okay, so it compares before and after the mutation, essentially. What kind of properties does it predict?
A whole range of really crucial stuff. Scientists sometimes call these modalities different types of biological information, so it can predict things like where exactly genes start and end, and this can differ between cell types. Where the gene sequence gets spliced, cut, and pasted together. Also, how much RNA is actually made from the gene. That's a measure of activity. And even things like which specific DNA bases are accessible or physically close to each other in 3D space, or which proteins are sticking to the DNA right there. Super detailed stuff.
And where does it learn all this from? It needs data, right?
Absolutely. Massive amounts of data. It was trained on information from huge public projects. You might have heard of some. ENCODE, GTEx, the 40 Nucleum Project, Phantom 5. These are big collaborations where scientists have experimentally measured all these properties in countless different cells and tissues. That's the ground truth the AI learns from.
Got it. So it learns from real-world biological data.
Yeah.
Can you give us a peek under the hood, technically? But, keep it simple.
Okay, simplified version. Think of it like this. It uses something called convolutional layers, which are great at spotting short, repeating patterns in the DNA, like little motifs. Then it uses transformers. You might have heard of those from language models. Here, they help the AI understand long-range connections, how parts of the DNA sequence far, far away might influence each other. That's critical for gene regulation.
Interesting, like understanding the whole paragraph, not just the word.
Exactly. And to crunch all this data, it runs on specialized hardware, these tensor processing units, TPUs, that Google developed, makes it efficient. And maybe one more point here. AlphaGenome builds on an earlier model called Informer, and it works alongside another tool, AlphaMessense. Alpha-missense is really good at the protein coding bits. That's only about 2% of our genome.
Only 2%.
Yep. AlphaGenome is designed for the other 98%, the so-called non-coding regions, which, it turns out, are incredibly important. There's like the conductors of the orchestra telling the genes when and where to play, and lots of disease-linked variants are hiding in there.
That 98% always sounded like junk DNA back in the day, but clearly not.
Definitely not junk. It's where a lot of the regulatory action is.
Okay, the mechanics make sense. But for me, the really interesting part is, what makes AlphaGenome stand out? Why is this the tool that might finally crack some of these big genetic puzzles that other tools couldn't? What's its secret sauce?
Yeah, great question. There are a few key things. First, and this is probably the biggest deal, it handles long sequence context at high resolution. So that million letters we mentioned. It analyzes that huge stretch, and it makes predictions down to the level of individual DNA letters, pinpoint accuracy.
Okay, why is that combo long and high-res so important?
Because gene regulation is complex. Sometimes the DNA element controlling a gene is really far away on the sequence, like thousands, even hundreds of thousands of letters away. Previous models, they usually had to choose. Either look at a long sequence but with blurry resolution, or look at a short sequence with high resolution. They couldn't really do both well.
Ah, like choosing between binoculars and a magnifying glass.
Kind of, yeah. AlphaGenome manages both. It can see the distant switches and exactly how they're wired up at the fine detail level. This lets it capture biological details others missed. Second big thing, comprehensive multimodal prediction. Because it handles long sequences at high res, it can predict the widest range of those biological modalities we talked about earlier.
So, more types of information from one tool.
Exactly. Scientists get a much more complete picture of what's happening around a gene, all from one place, not just fragments. Then there's the speed, efficient variant scoring. It doesn't just predict properties, it can figure out the impact of a specific genetic variant on all those properties incredibly quickly. Like, in a second.
In a second, for all that complexity.
Yep. It's built to efficiently compare the mutated versus the unmutated predictions and summarize the difference. That speed is huge for researchers testing many variants.
Okay, speed, range, detail.
Oh.
That's impressive. And you mentioned something specific about splicing earlier. Tell us more about that and why it's a breakthrough here.
Right, the novel splice junction modeling. This is really cool. So RNA splicing, think of it like editing a film. Before a gene's message, the RNA can be used to make a protein, certain bits need to be cut out precisely, and the remaining ends have to be stitched together perfectly.
And if the editing goes wrong?
Big problems. Tiny errors in splicing can garble the final message, leading to non-functional proteins. This is actually the cause of many rare genetic diseases. Think spinal muscular atrophy, some types of cystic fibrosis. They often involve splicing errors.
Okay, so it's critical. What's new with AlphaGenome?
For the first time, AlphaGenome can directly model from the DNA sequence alone exactly where these splice junctions are and how active they are, how much splicing is happening there. This gives researchers incredibly deep insights into how a specific DNA variant might mess up this crucial splicing process. It really opens up new ways to understand and maybe one day treat these kinds of diseases.
Wow, okay. That splicing insight sounds particularly powerful for certain conditions. So, impressive list of features. But, the real test is performance, isn't it? How does AlphaGenome actually stack up when you compare it to other tools already out there? Does it deliver in practice?
It really does. The results have been genuinely state-of-the-art. Let me give you some specifics. When they tested its ability to predict properties just from a DNA sequence, AlphaGenome did better than the best specialized external models in 22 out of 24 different tests.
Better than specialized tools.
In most cases, yes. And when predicting the effect of a genetic variant on regulation, it matched or beat the top external models in 24 out of 26 evaluations. And here's the kicker. Some of those external models were highly specialized, built just for one specific task. AlphaGenome matched or beat them while being able to predict all the different types of information modalities they tested. It was the only single model that could do that.
So it's not just broad, it's deep, too.
Exactly. It highlights its generality and power. There's a great quote about this from Dr. Caleb Leroux at Memorial Sloan Kettering. He said, "It's a milestone for the field. For the first time, we have a single model that unifies long-range context, base-level precision, and state-of-the-art performance across a whole spectrum of genomic tasks."
A milestone for the field. That's strong praise. Okay, so it performs incredibly well. But what's the actual benefit of having one unifying model like this instead of maybe using a collection of best-in-class specialized tools? Does it ever risk being a jack of all trades, master of none?
That's a fair question to ask, but the evidence so far suggests it's more like a master of many trades. The unifying aspect is actually a huge advantage for researchers. Think about it. Instead of juggling maybe 5 or 10 different specialized tools, each needing different inputs, maybe different expertise.
Right. Sounds complicated.
Exactly. With AlphaGenome, a scientist can investigate how one single variant impacts many different aspects of gene regulation, splicing, accessibility, protein binding, expression levels, all with a single API call. One query. This dramatically speeds up the process of forming hypotheses and testing them. It lowers the barrier to entry for exploring complex effects. Plus, it provides a really strong foundation for the wider community. Researchers can take AlphaGenome and fine-tune it on their own specific data or for their particular research question. It becomes a shared, powerful starting point.
Accelerating discovery across the board, potentially.
That's the hope. So let's pivot to those direct, real-world implications. Because this isn't just academic, right? It's about how this could actually impact health. First, disease understanding. AlphaGenome could really help scientists pinpoint the precise genetic causes of diseases much more accurately. Understand why a variant is causing problems. This could uncover completely new targets for therapies. It seems especially promising for those rare Mendelian disorders where one faulty gene has a huge impact. AlphaGenome could help find and understand those critical variants.
Makes sense. What else?
Second area, synthetic biology. Imagine designing artificial DNA sequences from scratch. AlphaGenome's predictions could guide engineers to create DNA that does exactly what they want it to.
Right.
Like, say, designing a sequence that only activates a therapeutic gene in specific cancer cells but leaves healthy cells alone, or activates it only in nerve cells, not muscle cells. Really precise control.
Wow. Targeted gene therapies designed with AI.
Potentially, yes. And third, just fundamental research. It could massively speed up our basic understanding of how the genome even works, mapping out all the important functional bits, defining what they do, especially in that vast non-coding space, helping us identify the absolute essential instructions for, say, making a liver cell function like a liver cell, illuminating the genome's dark matter.
Can you give us a concrete example of where it's already shown promise?
Yes, there's a great one involving a type of cancer called T-cell acute lymphoblastic leukemia, or TAL. Researchers had noticed specific mutations in the non-coding DNA of some TAL patients near a gene called TAL1, which is known to be involved in this cancer. They used AlphaGenome to predict the effect of these mutations. And guess what? AlphaGenome predicted these specific mutations would likely switch on the TAL1 gene by creating a new binding site for a protein called MYB right there in the non-coding region. This protein then helps activate TAL1, and activating TAL1 inappropriately is a known mechanism that drives this type of leukemia.
So the AI predicted a known cancer mechanism just from the DNA sequence change.
Exactly. It perfectly replicated what scientists already knew happened, showing its power to connect a specific non-coding variant directly to a disease outcome. Professor Mark Mansour at University College London summed it up well. He said, "AlphaGenome will be a powerful tool. Determining the relevance of different non-coding variants can be extremely challenging. This tool will provide a crucial piece of the puzzle, allowing better connections to understand diseases like cancer."
That TAL example really makes it concrete. The potential feels huge. But, okay, let's be balanced. Even groundbreaking tools have limits, right? What are the things AlphaGenome can't do yet? Where are the edges of its current capabilities?
Absolutely right. It's crucial to understand the limitations. So one challenge is still those very long-range interactions. We talked about it handling up to 1 million base pairs, which is great. But sometimes regulatory elements might be even further away, or their influence might be super subtle over those distances. Capturing that consistently is still tough. That's an active area of research.
Okay, so distance is still a frontier.
Yep. Another area is getting even better at capturing the nuances between different cell types and tissues. Gene regulation can be incredibly specific to the cell's context, and modeling all that fine-grained specificity perfectly is an ongoing goal. And this is really important. AlphaGenome has not been designed or validated for doing personal genome prediction. Meaning, you can't feed your whole genome sequence into it and expect it to reliably predict your personal risk for various diseases. That's not its purpose. Its focus is on characterizing the molecular impact of individual genetic variants, understanding the mechanism, not predicting overall individual risk based on the entire genome.
Got it. Molecular effects of specific changes, not a personal health forecast.
Precisely. And related to that, while it predicts molecular effects really well, like changes in RNA levels or protein binding, it doesn't give you the entire picture of how a genetic variation leads to a complex disease. Because diseases usually involve way more than just the initial molecular change, right? There are developmental factors, environmental influences, interactions between many genes. Those broader biological processes are beyond AlphaGenome's direct scope currently.
So it's a powerful piece of the puzzle, but not the whole puzzle itself.
A very powerful piece, yes. And the DeepMind team is very open about this. They're constantly working to improve the models, gather feedback, and push those boundaries further.
Good to know. So if researchers listening are excited by this, how can they actually get their hands on it? Is it available?
Yes, it is. AlphaGenome is now available for non-commercial research use through an API, basically a way for computer programs to talk to it. Again, the key emphasis is that the predictions are strictly for research use only. They haven't been designed or validated for any direct clinical applications, like diagnosing patients or guiding treatment decisions right now.
Research tool, not clinical tool.
Yes.
Understood.
Correct. But the team really wants researchers globally to use it. They're inviting people to get in touch with ideas for how AlphaGenome could help their specific research. And they've set up a community forum for questions and feedback. The aim is definitely to get this powerful tool into the hands of scientists who can use it to make discoveries, to deepen our understanding of, how life is encoded.
Fantastic. That accessibility is key. So just to kind of wrap our heads around this, AlphaGenome feels like this incredibly powerful new lens, maybe the most powerful yet, for peering into our genetic blueprint, helping scientists finally make sense of those complex instructions. It seems poised to really speed up discoveries, especially around diseases, by untangling how these subtle DNA variations work, particularly in those huge non-coding regions that were so mysterious before.
Yeah.
So what does this mean for you listening in today? Well, it means you're now clued into a really significant scientific advance, one that could genuinely change human health down the line. From understanding why diseases happen to maybe even designing smarter therapies. It's a fantastic example of how AI is helping us tackle problems that seemed almost impossible just a short while ago, peeling back the layers of our own biology. Okay, so here's a final thought to leave you with, something to chew on. We've talked about AlphaGenome decoding gene regulation. What other massive, long-standing mysteries in biology do you think we could unlock if we could build AI that truly understood every single instruction in our cellular manual, every interaction, every nuance? Keep that question percolating and keep diving deep with us into new knowledge.